Gene editing is revolutionizing the way we think about genetics and health. Imagine being able to precisely alter DNA to eliminate genetic disorders or enhance crop resilience. Techniques like CRISPR allow scientists to snip out faulty genes and replace them with healthy ones, almost like editing a document. This technology has the potential to eradicate diseases like sickle cell anemia and even tackle complex issues like climate change by creating more robust plants. However, with great power comes great responsibility. Ethical concerns arise about designer babies and unintended consequences in ecosystems. As we stand on the brink of this genetic frontier, the question remains, how far should we go in rewriting the code of life? The possibilities are endless, but so are the implications. Gene editing is a powerful tool that allows scientists to make precise changes to an organism's DNA. Here are some key aspects of gene editing. CRISPR-Cas9 is a popular gene editing tool that uses a small RNA molecule to locate a specific sequence of DNA and then cuts the DNA at that site. TALENs is a gene editing tool that uses a DNA-binding protein to locate a specific sequence of DNA and then cuts the DNA at that site. ZFNs is a gene editing tool that uses a DNA-binding protein to locate a specific sequence of DNA and then cuts the DNA at that site. Applications of gene editing include treatment of genetic diseases, where gene editing can be used to correct genetic mutations that cause inherited diseases, cancer treatment, where gene editing can be used to selectively kill cancer cells by disrupting genes that are specific to cancer, gene therapy, where gene editing can be used to introduce healthy copies of a gene into cells to replace faulty or missing genes, agriculture, where gene editing can be used to introduce desirable traits into crops, such as resistance to pests or diseases, and synthetic biology, where gene editing can be used to design and construct new biological systems, such as microorganisms, to produce biofuels or clean up environmental pollutants. The benefits of gene editing include precise control, allowing scientists to make precise changes to an organism's DNA, efficiency, as gene editing can be more efficient than traditional breeding techniques, reduced risk, as gene editing can reduce the risk of introducing unintended traits into an organism, increased crop yields, as gene editing can be used to introduce desirable traits into crops, leading to increased yields, and new medical treatments, as gene editing can be used to develop new treatments for genetic diseases. However, there are challenges and concerns, such as off-target effects, where gene editing can sometimes introduce unintended changes to an organism's DNA, mosaicism, where gene editing can sometimes result in a mixture of edited and unedited cells, germline editing, where gene editing can sometimes introduce changes that are passed on to future generations.